My name is Mathias Ritzmann. I'm working at the veterinary faculty at the Munich University in Munich as full professor for swine diseases, and I'm also head of the clinic for swine at the university in Munich. I'm going to talk about different PCV2 associated diseases and the possibilities of control of, for example, PMWS with a focus on the vaccines applied to the piglets.
Ni hao. Porcine circovirus can cause different diseases and is linked to different syndromes in pigs. The most important one is still the PMWS, but we also know the link to the PDNS. We know that PCV2 can cause lung diseases, or the so-called involvement in the PRDC. We know that PCV2 can cause reproductive disorders, PCV2-RD. It can cause enteric diseases, and we also have to consider that a subclinical infection can occur. That's the so-called PCV2-SI. But the most important one, I think, it's still the PMWS or the systemic disease. If we think about diagnosis of PCV2-associated diseases, we have to consider that the clinical signs are in some cases unspecific. So if we think about the PMWS, we know that wasting or coughing or other signs which we can see in the PMWS are nonspecific and can also occur in different other diseases. So it's necessary to do a necropsy in the pigs. Picture of one of our pathologists working in swine diseases. And we have also to detect the pathogen, for example, with different methods, like the qPCR, like the immunohistochemistry, or the in situ hybridization. So we think a link between all 3 points is still necessary. So the clinical investigation in the farms with perhaps non-specific signs, the necropsy, and the detection of the pathogen with different methods. We also know that PCV2 is associated with complex diseases. For example, the porcine respiratory disease complex. So that's a combination of infectious and non-infectious factors which can occur within a farm. And PCV2 might be one of these infectious factors. But we also know that other pathogens like Mycoplasma pneumoniae, like influenza virus, like PRRS virus, like Haemophilus parasuis, or also APP may be involved in a PRDC. And we also know that the environment and the housing conditions may play a role in the clinical outcome of a PRDC. And there are lots of different studies showing this synergism or association between PCV2 and other pathogens or within other pathogens. There are lots of studies coming from all over the world, and also pretty good and pretty new studies coming from China showing these association and this synergism. So, for example, we have a strong link between a PCV2 infection and a PRRS infection, and the same could be seen between a PCV2 infection and Mycoplasma pneumoniae. And if you think about the virus, the first link was in the 90s, where PCV2 was considered to be a pathogen of the clinical outcome, the wasting syndrome, later called as PMWS. And at that time, we didn't know very much about the virus. And with the beginning of the year 2003 or 2004, we have seen the link between different genotypes within porcine circovirus type 2. And at that time, we think that the PCV2A played the major role in the clinical outcome of the infection. So one example from an older study which we have done, where we have seen the shift to the PCV2B. This started in the year perhaps 2008, 2009. So these are different farms in Germany where we have investigated the piglets at different ages. So with the beginning of week 8, week 12, week 16 until slaughter in week 24. And the blue bars are showing the percentage of PCV2B positive pigs. The red bars are showing PCV2A. And in single pigs, we can detect both, so PCV2A and PCV2B. So some years ago, we have seen mainly the PCV2B infection in the pigs. And since a couple of years, I think it started 2009 in some areas of the world, or 2012, a little bit later in Europe, we have seen the shift from PCV2B to PCV2D. And if you look at the literature, there are lots of pretty good and pretty new studies coming from China showing that the predominant genotype is nowadays the PCV2D. And if you look at the abstract book, there are lots of presentations tomorrow and on Monday also with the topic of the PCV2D. These are some results from our own studies where we have investigated different pigs with typical clinical signs of PMWS or PDNS. And in more than 60% of the pigs, we have found the PCV2D. In about 30%, we have seen that PCV2B is present. And in just 7% of the cases, we have seen PCV2A infection. So I think that's a situation which is pretty similar to other areas like to US or especially like to Asia, also in China, that we mainly are now finding the PCV2D genotype. And if you think about control of PMWS, we know that the improvement of the farm management and farm hygiene has a positive effect. So lots of years ago, before we had vaccines against PCV2, the 20-point rules, according to MADEC, was pretty popular all over the world. And these rules showed clearly the impact of non-infectious factors to improve the health situation in the pigs in the farms. Since a couple of years, we have the vaccines against PCV2, which are used all over the world. There are still some topics we are working about it. One point I want to go a little bit more in detail, that's the effect of the vaccination against PCV2. So this is a study which we have done for a couple of years, where we vaccinated the pigs with an age of 3 weeks. And on the other hand, we have non-vaccinated piglets. So the green bars are showing the vaccinated pigs, and the red bars are showing the non-vaccinated pigs. And we have looked in the blood samples when viremia started. And in this farm, viremia started with an age of about 7 to 10 weeks. So if you look at the clinical results showing on the left-hand side before the viremia started, we didn't see any differences between the vaccinated and the non-vaccinated pigs. That's easy to explain because the pigs were not positive for PCV2. So we have seen no differences between coughing or other respiratory signs like dyspnea or also like fever. And when viremia started in week 7 to 10 in this farm, and we look at the results of the PCV2 vaccination, we have seen significant differences between the vaccinated and the non-vaccinated group. So a picture on the right-hand side shows coughing pigs in the vaccinated group with lower than 30% and more than 40% in the non-vaccinated group. And other respiratory signs, it's about 3% in the vaccinated group and nearly 10% in a non-vaccinated group. So if we just look at the clinical outcome of a PCV2 vaccination, we have to consider when is the time point of infection. That's the same farm showing other results. On the left-hand side, that's the mortality. That's a mortality calculated from the vaccination until slaughter. So in the vaccinated pigs, we have about 5% mortality from vaccination until slaughter. And in a non-vaccinated group, that's a red barn, we have about 10% mortality. So mortality is double as high as in the vaccinated group. And picture on the right-hand side shows the number of wasting pigs or runts. We have defined them as 25% lower body weight in the corresponding group. So if we look at week 12, we have had about 6% of wasting pigs in the vaccinated group, and we have more than 12% of wasting pigs in the non-vaccinated group. Same situation in week 17, and also the same situation just before slaughter in week 23. So about 2% wasting pigs in the vaccinated group and nearly 5% in the non-vaccinated group. And if you look at viremia in these pigs, that's the same trial. There you can see the green line, that's the vaccinated group, and the red line, that's the non-vaccinated group. So viremia starts with about an age of 7 weeks of life, and we see clear differences and also significant differences between the vaccinated group and the non-vaccinated group. So the duration of viremia in mean is about 30 days longer in the non-vaccinated group compared to the vaccinated group. And the end of the viremia is in mean about 27 days later in the control group compared to the vaccinated group. But we also have to consider that we can find PCV2 by PCR in the vaccinated piglets. That doesn't mean that they are non-protected. We can also find PCV2 in fully protected pigs. We know this from the infection trials. But we see a lower percentage of positive pigs and a lower number of pigs with a high amount of PCV2. That's shown in this trial. That's another farm where I've also tested a vaccine and have a control group, that's the right-hand side, and a vaccinated group. And the pigs were also vaccinated with 3 weeks of age. And if you look at the orange barns, these are the amounts of PCV2 measured by the PCR in the blood samples with more than 10^6 copies per ml. And the brown barns are PCV2 amounts also measured by the PCR with more than 10^4 copies per ml. And if you compare the vaccinated group with the non-vaccinated group, you see clearly differences. So also shorter viremia shown on the left-hand side, longer viremia shown on the right-hand side, a lower number of positive pigs with a lower amount, so that's 10^4 up to 10^6, and especially a lower number of pigs which have the high amount with more than 10^6. So by the same result, we also can find PCV2 by PCR in vaccinated pigs. These are 6 studies which we have done where we had a control group and a vaccinated group showing the differences in the average daily weight gain. And we have farms in Germany with a lower average daily weight gain, and we have farms with a pretty high average daily weight gain. This might depend on genetics, on a feeding system, on other diseases within the farm. But if you look at the results, we have seen in every farm where we have vaccinated the piglets with 2 or 3 weeks of age, a higher average daily weight gain from beginning of the fattening, usually week 10 or 11 until slaughter, usually week 21 to 22 or 23. So it doesn't matter if this is a farm with a lower average daily weight gain or a high average daily weight gain, we see the clear effect of the vaccination of the piglets within these farms when we are vaccinating them with 2 or 3 weeks of age. And in one of these farms, we have looked for the co-infections. So green, once again, these are the vaccinated piglets, and red are the non-vaccinated piglets. And if you look just at the detection of PCV2 in the lymph nodes, I think that's clear that in the vaccinated piglets, the percentage of positive pigs is lower in the vaccinated group. But we have also seen an effect on other pathogens showing in this slide. So we have a lower detection rate of Porcine virus in the lungs when we have vaccinated the piglets against PCV2 compared to the control group. We have also seen a lower detection rate of Mycoplasma pneumonia in the vaccinated pigs, PCV2 vaccinated pigs, compared to the non-vaccinated pigs. And also a significant lower detection rate in Mycoplasma harrowinus. So we also know that PCV2 vaccination has an influence on other pathogens within the farm. We have a pretty strong discussion about the usage of antibiotics in Europe. I think you have heard this morning some aspects of the discussion about the antibiotics worldwide. And in Germany, we have a law where we have to reduce the antibiotics in farm animals. And that's a trial we have done. We have had a control group, so non-PCV2 vaccinated pigs, where we have a group where the pigs were vaccinated in the first week of life, and we have had a group where the pigs were vaccinated in the third week of life. And we have counted the number of treatments with different antibiotics with different syndromes. So, for example, if you look at the skin diseases, in the control group, 23 pigs were treated with antibiotics. In the group which was vaccinated in the first week of life against PCV2, 5 pigs were treated. And in the group which was vaccinated in the third week of life, only 4 pigs were treated. And if we count them together, or look at the number of percent treated pigs, we can see clear differences. So in a non-vaccinated group, 90% of the pigs were treated with antibiotics during this trial. In the group which was vaccinated in the first week of life, only 15% was treated. And if you look at the pigs which were vaccinated in the third week of life against PCV2, only 11% was treated. So also clear reduction of the usage of antibiotics when we are vaccinating the piglets. And we have seen the better effects when we were vaccinating them with 3 weeks of age compared to 1 week. Second point I want to go a little bit more in detail is the point of time of vaccination against PCV2. These are 2 farms in the southern part of Germany where the piglets were non-vaccinated. And in the farm on the left-hand side, the sows were vaccinated against PCV2. And we have measured the IgG and the IgM in the blood sample of the pigs, beginning from the first week of life until slaughter in week 26. And on the left-hand side, you see a pretty high number of positive samples in the pigs for the IgG. These are maternally derived antibodies. We do not see pretty often this clear drop down in week 7. So usually we see a slight decrease of a number of positive pigs regarding the IgG. And the picture on the right-hand side is pretty much more often, so lower percentage of positive pigs due to the MDAs, and an increase of the IgM starting in week 7. That's due to the infection because the pigs were non-vaccinated. And on the left-hand side, where the pigs were also not vaccinated, but the sows were vaccinated, we see a little bit later the increase of the IgM. So a later detection of the IgM and the IgG, but we can see the infection during the fattening period. So we couldn't show that the vaccination of the sows alone could prevent the pigs from infection. And these are 5 farms where we have measured blood samples with qPCR, and these results are showing the amounts with more than 10 to 6 copies per ml. And we see clear differences between different farms in Germany. So we have some farms with a pretty late infection. So if you look at the green line, the infection starts in week 14. So that's pretty late, mid of fattening, where the infection of PCV2 takes place. We can see in most of our farms that infection is during the nursery. And in most of our farms, we see the infection not before week 6 or 7 of life. But we have to consider that the time point of infection differs between the different farms. And in this point, we have also to consider the state of the sows, if they are vaccinated against PCV2 or none. That's another trial where we have had non-vaccinated piglets, where we have vaccinated the sows and the piglets are non-vaccinated. And the green bars are showing the results of the piglets which were vaccinated with 3 weeks of age. And if you look at the PCR results, the copies per ml in the blood samples, and if you look at the non-vaccinated pigs, these are the red bars, and the results of the piglets where their sows were vaccinated but not the piglets, there are just slight differences. But if you look at the piglets which were vaccinated, we have still some single positive pigs. I think that's normal. But we can see the clear differences between the non-vaccinated pigs and the pigs where the sows were vaccinated and pigs were non-vaccinated. And if you look at the higher amount, so that's more than 10 to the 6 up to 10 to the 8, we have no pig which was positive, which was vaccinated with 3 weeks of age. But we have positive pigs which were non-vaccinated and pigs where the sows were vaccinated. So especially in the clinically relevant amounts, more than 10 to the 6, or might be more than 10 to the 7, there's a clear reduction of positive pigs. That's the same trial which we have done in another farm, showing a little bit different results. So on the left-hand side, there is are piglets coming from sows which were vaccinated against PCV2, and we have taken the blood samples with the beginning of week 12 until slaughter. And each dot represents a single pig. And on the right-hand side, these are piglets which were vaccinated with 3 weeks of age. And the results are pretty similar if we compare to the study which we have done before. So lots of pigs which are positive when the sows are vaccinated, and just single pigs, which was just one pig in this farm, which is positive when the piglets are vaccinated. And if you look at the average daily weight gain at the viremia of these pigs, we see nearly no differences in week 13 of life, so that's about 3 weeks after beginning of fattening. We see slight differences in week 16, but we can see significant differences in week 19 and week 22 in the number of positive pigs. So nearly the same results as in the studies which we have seen before. And if you look at the average daily weight gain of these pigs, so comparison of piglets which were vaccinated, these are the yellow bars, and of piglets where the sows were vaccinated but the piglets are non-vaccinated, we have seen at the end of slaughter significant differences. So piglets which were vaccinated had average daily weight gain of 762 grams per day, and piglets which were non-vaccinated but the sows were vaccinated had an average daily weight gain of about 736 grams. The next point I want to go a little bit more in detail is the discussion about an influence of maternally derived antibodies. These are 2 trials which we have carried out. One farm is located in the southern part of Germany, that's the left-hand side, and one farm is located in the northern part of Germany, that's the right-hand side. And there we have also non-vaccinated piglets, that's the red bar. We have piglets which are vaccinated in the first week of life, that's the light blue bar, and we have piglets which were vaccinated in the 3rd week of life, that's a dark blue bar. And if you compare the results of the non-vaccinated piglets and the piglets where the sows were vaccinated against PCV2, we see nearly no differences. So no impact on the average daily weight gain when only the sows are vaccinated. But we can see a clear influence on the average daily weight gain when we are vaccinating the piglets 2 weeks later, so in the 3rd week of life. And there we've had, so as well as in the southern part of Germany and in the northern part of Germany, significant differences between the groups which were vaccinated in the first week of life and the piglets which were vaccinated in the third week of life. And then we have looked at the blood samples of these pigs. So we've taken lots of blood samples beginning from week 1 until slaughter. The graphs on the left-hand side show the IgG, and the graphs on the right-hand side show the IgM. The dotted line is the control group. The dashed line are the pigs which were vaccinated in the first week of life, and the drawn through line are the piglets which were vaccinated with 3 weeks of life. And if you just look at the IgG, we see nearly no differences between the groups. And if you go back to the average daily weight gain of these farms, these are the pigs from these farms, we see clear differences in the average daily weight gain, but nearly no differences in the IgG. So if there is an influence of MDAs, it might be independent on the number of positive pigs regarding the IgG. And if you look at the IgM on the right-hand side and look at the drawn through line, we can see that the piglets which were vaccinated with 3 weeks of life had an increase of the IgM after the vaccination, but we couldn't see this increase in the piglets which were vaccinated in the first week of life. And in another trial, we have measured the duration of viremia. And there we have divided the pigs with low antibody titers, that's lower than 1/100 measured by the IVAD, and pigs with a high antibody titer. The red bar is showing the control group, which is non-vaccinated. There we have about 60 days viremia. And if you look at the piglets with a low antibody titer at time point of vaccination, we have seen that the viremia is about 30 days, and it's about 1 week longer in the piglets with a high antibody titer. But if you look at the number of the pigs, you see that just a few of them had the high titer, and most of the pigs had the low titer. And between the groups of with the low titer and the high titer, there were no statistically significant differences, but a slight increase of the duration of the viremia. And if you compare the average daily weight gain of these pigs, we have calculated the difference between the control group and the group with the low titer and the group with a high titer. So for example, the control group, the non-vaccinated group, was slaughtered with 100 kilogram body weight. So we have seen in the pigs with the low titer 4.7 kilogram more compared to the control group, and in the group with a high titer about 3.6 kilogram more compared to the control group. These differences are non-significantly different, but we can see a slight influence on the titer. And we know from other studies from Europe, one study coming from Italy, from Professor Mathias from Parma University, and he has seen when sows are vaccinated against PCV2, the vaccination of the piglets with 6 weeks of age is more effective for controlling natural PCV2 infection compared to the vaccination of the piglets with 4 weeks of age. So, small conclusion of the MDAs, we know that there is an influence when we are vaccinating the piglets in the first week of life. We have nearly no influence when we are vaccinating the piglets with 3 weeks of age. Lots of results are showing also 2 weeks of age. But it has to be considered if the sows are vaccinated against PCV2. So perhaps in this case, we can discuss if it makes sense to vaccinate the piglets a little bit later. And we have had in some countries in Europe, like in Denmark, like in Spain, also in Germany, the discussion if it's possible to eliminate PCV2. So we have lots of experience with the elimination of Porcine virus. We have experience with other pathogens like Mycoplasma, and the same questions came out some years ago if it's possible to eliminate PCV2 under farm conditions. Just one farm with single results. So when we started vaccination in the piglets with 3 weeks of age, we have seen the same result like in lots of other studies which we have done. So single pigs are positive with more than 10 to the 6 copies per ml, and some pigs are positive with more than 10 to 4 copies per ml. And when we look into the pigs about 10 weeks later, we can see the clear effect of the PCV2 vaccination. So a lower number of pigs which are positive, especially for the high amounts of PCV2 detected by the PCR, and shorter time of the duration of the viremia. So I think that's a result which we have seen in lots of other studies. And in this farm, we have taken blood samples half a year later, shown in this slide, and we've also taken blood samples 1 year later when we started with the vaccination in this farm. And you can still find positive pigs. Number of pigs is still going down, but in our opinion, it's not positive. It's not possible to get rid of PCV2. So in every farm which we know, we can find PCV2, and in our opinion, it's not possible under normal farm conditions to eliminate PCV2. So to summarize, we know lots of positive effects of the vaccination of piglets against PCV2. For example, the higher average daily weight gain or the higher body weight at slaughter. We see the decrease of the clinical signs, like the respiratory signs. We see the clear decrease of number of wasting pigs or runts, and also the mortality. We can see the reduction of viremia, and also the reduction of the co-infections like virus, like mycoplasma, high pneumonia. And we have also seen in lots of our farms in Europe that we can reduce the usage of antibiotics in farms which are vaccinated against PCV2. In our opinion, elimination of PCV2 under field conditions is not possible. And if we think about control of PCVD, we have to improve farm management and farm hygiene. I think we have all over the world farms with a pretty good farm management, a pretty good farm hygiene. But I think we also have still farms all over the world where farm management, farm hygiene is not perfect. And we have the possibility to vaccinate against PCV2 in sows and or in piglets. So for vaccination against PCV2 in Europe, it's usually carried out with 3 weeks of age, with some farms where the piglets are vaccinated with 2 weeks of age. And if you think about the influence of MDA, I think there is an influence when we're vaccinating the piglets in the first week of life, but we have no bad effect when we are vaccinating the piglets with 3 or 2 weeks of life. But under some circumstances, as I've mentioned before, especially when we think about the vaccination of the sows, we can consider if it's possible or makes sense in some farms to vaccinate the piglets later, for example, 5 weeks or 6 weeks of age. And with a picture of my colleagues from the Clinic for Swine, I'd like to thank you for the attendance, for listening, and I think we have lots of time for questions. So thank you once again.